Great. Well, good morning, everybody. It's an absolute pleasure to be here with all of you. Thank you for joining this webinar, which is all about decoding soil biology for soil health. This webinar has been organised by Soil Association, a UK charity that's worked to transform the way we eat, farm and care for our natural world for nearly the past 80 years. The Soil Association has been working in collaboration with 26 other partner organisations across the whole of Europe in this EU Horizon funded project, AI for Soil Health. My name's Joseph Gridley. I work in part of the Soil Association. I run a business there called Soil Association Exchange, and what we do is help farmers all over Europe to measure their impact on the environment and then get the advice and financial incentives that they need to improve. The biological indicators we look at in Exchange are earthworms. We find it's a really practical and great way for a farmer to start to understand the biology in their soils and what they can do more. Before I properly introduce today's session, just a few bits of housekeeping. Today's webinar is being recorded. We'll share a link with you after it's all done, so you can go back, share it with colleagues, watch it again if you wish. The format of today's event is going to go as follows. You'll have a short introduction from me, and then our three speakers, Lure, Sonia, and Maeva, are going to give three quick presentations, and then we'll get into some question and answer. Throughout the whole session, we'll be using Menti. You can see here at the top of the screen, menti.com. If you go to menti.com and put in that code, you'll see lots of questions as we go through to give you a chance to engage and be part of this conversation too. We'll also invite your comments and questions. So if you look right down at the bottom of your screen, you'll see the Q&A chat box. If you've got a question for any of our speakers, for me, at any point, just type it in that question and answer box, and we'll try and get as many of those questions answered during this session. Also, feel free to introduce yourselves in the chat. I'm always amazed in these meetings how we've got people from literally every corner of the world, amazing expertise. So please, do say hello, say who you are, where you work, why you're interested to be here in the chat. Maddie is on hand, so if anyone's got any technical difficulties, stick something in the chat and she'll be there to try and help. So, why are we all here? AI for soil health. I mean, I'm sure it comes as no surprise to many of you, but the health of soils across Europe are under threat. A transition to a sustainable future is possible, but the risk for taking these changes, for transitioning, sits firmly on the shoulders of farmers, and it's risky business. This is new stuff for them. And so we really need to understand what practices work for farmers, which ones don't, and how can we protect the environment and protect farm businesses, make them more resilient in the face of climate change. The AI for Soil Health project has been funded by Horizon Europe, and it's building an AI-powered soil health monitoring system to accelerate the collection and use of soil information. We aim to help farmers and land managers measure soil health without the need of laboratories. We do a lot with laboratories in Exchange, and it's expensive and it's slow. And so how do we roll this out to all farmers? We want to give policymakers, people in government, the ability to have the information to know what's working out on farms and what's not. The climate crisis is here already. In the UK alone, in the last 3 years, we've had the hottest and driest summer, the wettest autumn, and then the warmest and driest spring on record. This is already here, it's already coming for us, and we need to understand our soil so we can help farmers navigate this complicated future. AI for soil health is at the forefront of working with stakeholders to co-design, create and harness digital technology to support pan-European efforts to halt and reverse soil health decline. We want a world where every farmer can do their own on-farm experimentation into soil health. From July to November, this project has been hosting the Envisioning the Future of Soil Health Monitoring series. It's been a series of 5 online events, each focused on a single hot topic at the intersection of soil science, digital innovation, and EU policy. Today is the 5th and the final one in this series. So welcome to today's webinar. It's all about decoding the biology of soil health. We're joined by AI for Soil Health project partners and experts to discuss the latest research and innovations in decoding soil biology, from measuring microbial diversity and enzyme activity to transforming biological data into practical tools for soil health management. The panel will explore how biological indicators can enhance soil monitoring, how technology is bridging the gap between science and farm management, and what large-scale studies reveal about the relationships between soil life, land use, and climate in Europe. But before I introduce our amazing speakers, we've got some questions for you. So as I said earlier, if you go to menti.com, you can do it on your phone, you can do it on your desktop, and put in that code, or you can just scan that QR code as well. We're going to ask a couple of questions for you to see who we've got here in the room with us today. Are you all ready? Got that done? Everyone on the Menti? I can see that number going up. Perfect. So first question, we want to learn what skill sets we've got in the room. So select the ones that are most relevant for you, but what are your domains of expertise? What fields are you working in? What kind of stuff are you doing in your day job? It'd be great to understand where this group sits. Amazing. Lots in the soil health science. No big surprise there. I guess it's on the title of the webinar, but great to see that we've got some data science and some AI people too. Seems like we've got, land use and management practice people are taking the lead here. Lots of people with practical agricultural expertise, which is great to see. And got some good recognition from people who are working more in the policy space too. Okay, so next question, we want to understand at what scale are you working? Are you doing stuff in a local region of your country? Are you doing it at a regional level, a national level? Maybe you're working for soil health across the whole of Spain or the UK, or maybe some of you are going pan-European. Can we solve this challenge across all 27 member countries in the EU? National seems to be taking the lead, but we've got a good contingent of those doing this at a continental level, and then a good number at the local. And finally, it would be great for us to understand why you're here today. What is it that's made you turn up, clear your diary for this hour and a half to come and learn more about soil biology? What is it that you hope to get out of today? Just scribble down anything that comes to mind, but we're really keen to understand your motivations. Super. Looking for inspiration. That's always good on a Monday morning in the middle of winter, I find. Uh, Knowledge, learning more about soil biology and AI, you'll definitely get that. Soil-borne plant disease management, yes. Latest research, how can I improve my farm soil? That's great. There'll definitely be some applied bits today, stuff that you can take back to your farm. Current trends, what's relevant for other stakeholders. How this links through to policy. I think that's going to be a real key topic that hopefully we can unpack in today's conversation as well. That's great as well. I have a 2-year-old son, and he always wonders what I do, and I try and explain to him it's all to do with soil health. So hopefully, today's presentations are going to be in a way that we can take back to share with young people, so that they can keep protecting years to come too. Amazing. Well, thank you very much. It's great to have you all here, and we'll try and do all of those. So before I introduce our first speaker, Le, we're going to do a quick quiz to see your starting point of knowledge on all things soil and biology. So question for all of you. What percentage of the planet's biodiversity lives in soil? 0 to 44%, 45 to 74%, or 75 to 99%. Make your votes now. You can see a little bit on the right. Here are some pictures of some of the things that are living in our soil. Super. We've got a few more people putting their votes in. Okay, that's question one. Question two, how many bacterial and archaeal cells can be found in a single gram of surface soil? Less than 100,000, more than 1 million, or more than 1 billion. So In a single spoon of soil, how many cells can you find? More than a billion, more than a million, less than 100,000. Seems like we've got a smart group on our hands, Lure. No one thinks it's less than 100,000. And final question before we hand over to Lure. What is the largest organism in the world? Great Barrier Reef, a giant sequoia tree, a pando, a clonal aspen grove, the Armillaria austriae honey fungus, or the blue whale. Amazing. So no one's going for the top three options. We seem to have a lot of people that are interested in this honey fungus, a few on the blue whale category. There is another option, which is me after a really big lunch on a Sunday. I definitely probably could rival a blue whale at that point. Wonderful. Well, Lure, I know you're going to tell us the answers. But just before we get into your presentation, a small few words of introduction. Dr. Lure Pelde, she's a biologist specializing in soil microbial ecology. She's based at Naker in the Basque Country. That's the top bit of Spain where it rains lots. Beautiful, beautiful grassland and great food. And she's a researcher in the Department of Natural Resource Conservation and coordinates the Soil Microbial Ecology Group. She's done amazing research all over the world. And her work is really on assessing soil health and testing management strategies aimed at restoring soil biodiversity and its associated functions. To this end, she explores the potential of microbial communities as indicators and develops methodologies ranging from advanced omics techniques to simple in situ applicable tools. So, Lure, it's an absolute pleasure to have you here today. Over to you.
Okay, thank you. Can you hear me and see the screen?
Loud and clear.
Yeah, perfect. Okay, so I will start with the answers to the questions in the Menti, even if, we see that there are lots of experts around today. So what fraction of the planet biodiversity lives in soil? It's not over 75%, but a big amount, so around 60% of life lives in soil. So it's our most biodiverse habitat. Some of these creatures can be seen without a microscope, but most of them are microscopic, are microbes, fungi, bacteria, archaea, and these are precisely the ones that cover or make the soil function, are in charge of the main geochemical processes. The second question, most of you answered it well. So a gram of soil can contain more than 10 to 9 bacterial and archaea cells, but also trillions of viruses, tens of thousands of protists, and 200 meters of fungal hyphae. So it's amazing. And no, it's not the blue whale. The largest organism in the world is the Armillaria austriae, a fungi that covers an area of up to 10 square kilometers in Oregon, and it weighs around 200 whales, so blue whales. So it's really amazing. And all this amazing biodiversity is a nature-based solution that provides or supports the provision of functions and services by the soil. So covering a wide variety of functions like supporting nutrient cycling, plant growth and productivity, enhancing water retention and purification, also improving resilience against pests and diseases, helping to mitigate and adapt to climate change, and so on and so on. So as an example of this link between soil biodiversity and health, I've brought this publication that they developed an index based on some biodiversity metrics, and they found it was related to one of the key supporting services provided by soil, which is productivity, primary productivity. They also found positive association between some taxa and the productivity, and between some functional groups and the productivity, which I find really interesting. For example, when I hear people say that soil biology is too complex and hard to interpret, so we cannot use it for measuring soil health. This is, for example, a key link or a clear example that we can link nitrogen-fixing bacteria or mycorrhizal fungi positively to productivity, and instead plant pathogens, we can link them negatively to productivity. It's also interesting to see that there are more and more efforts related to linking soil health and human health under this paradigm of ecosystem, well, of One Health, linking soil microbiome diversity and our gut microbiome diversity. And I think in the following years, this will provide a clear support to soil biodiversity studies. But despite all of this, if we have a look at the frequency of different biological indicators in soil health assessment approaches, we can see in this graph, you see the biological measurements in green, and you see that they are very few, and their frequency is also quite low. So at least until now, the reuse is not very widespread. Maybe now that we have the soil monitoring law that is requiring the measurement of DNA metabarcoding, things might start to change. And why is this? Why these biological measurements are not used so routinely? They have some advantages that I have talked about. They are ecologically relevant, they have an integrative character, they are sensitive to changes in management, but they also have some challenges. They have a lack of harmonized methodologies, they require resources and specialized skills, and they also have a high temporal variability. And what are we doing in AI for Soil Health project to tackle these challenges? So among other efforts, we have a network of 13 pilot sites covering different countries, different land uses and management for climatic conditions. And we are using these sites for testing and validation purposes. What for? For example, I have mentioned you that soil biology often requires specialized labs and experts to interpret the results. Well, maybe not anymore, because Sonia will explain you later the equipment they are developing to measure enzyme activities in soil. There are also other commercial initiatives like microbiometer, a test that measures microbial biomass and fungal to bacterial ratio in an easy way, or the Soil Health Institute is promoting this SLAKES app that tries to measure aggregate stability using a mobile phone. So we are, in the project, we are using these methods, and we are comparing them to traditional lab-based methods to see if they correlate well or not. Another of the challenges that I have mentioned you is that the biological properties are very sensitive, which is good in some sense, but they could be too sensitive, so they are temporally very dynamic. And to test this, we have used the Spanish pilot site, which is an experimental grassland where sheep are grazing, and we have monitored from the whole grazing season, from April to November, different.
Lure,
Yeah.
sorry to interrupt you for just 1 second.
Yes.
There's a little box over your graph. Could you click the occultar bit,
Oh, sorry.
and then everyone can see it?
No.
Perfect. Thank you.
Yeah, I'm sorry. Okay. Yeah. I was saying that we have done this temporal study where you can see in red the temperature and in blue the humidity of the soil throughout this April to October time frame, and also some biological properties like microbial biomass carbon, enzyme activities, and potentially mineralizable nitrogen. And you can see that there is a variation in time, so they tend to lower the values in summer. They tend to recover again in autumn. And we have also measured in this same study metabarcoding of bacteria and fungi. At least at the phylum level, it seems that the variation is not so high as with the other biomass and activity parameters. And we also have to take into account that this variation may come also from the spatial heterogeneity of the soil, because we tried to go to the same, to sample the same spot again, but obviously the same soil is not there anymore. And for example, I show you the values of the clay percentage, which obviously they are not changing in such a short time, but there is also a certain variability from sampling time to sampling time. Finally, we have also measured the biodiversity profile, but at the RNA level, which is the fraction, this fraction should be more related to the active microbial population. And in this profile, the variability is much higher. So with all of this, we are trying to think about which microbial parameters are more appropriate for which purposes and contexts. For example, in a national level, where we are interested in large-scale monitoring and long-term monitoring, maybe the most sensitive and variable properties wouldn't be the most appropriate ones. So this is what we are trying to think about with these studies. And finally, we are also using this data that we have generated in this pilot site to try to work also on emergent properties that enable a systemic assessment of soil health. In this case, we have analyzed, or we are creating networks to gain understanding in terms of organization. So not only biodiversity, but also the complexity of the communities in the soil. How are they related to each other, the individuals living in that soil? So this is very briefly what's going on in the project. It's still an ongoing study. And yeah, just to finish, I would like to mention a poem in Spanish by Machado, which says, "Caminante no hay camino, se hace camino al andar," which means that there is no path. The path is made by walking, and this is the path we are trying to walk, trying to somehow mainstream the use of soil biological properties in soil health evaluation. So that's it from my side. Thank you.
Wonderful. Thank you so much, Lur. An amazing presentation. Uh, and what it really kind of brings home to me is how important, I guess, all of these biological indicators are, but the inherent complexity that sits there, depending on the temperature, the rainfall, the type of soil, the time of year. But like you say, we need to spend more of our time understanding these things because they're such important properties to a soil's health. So thank you. So it's back to you in the audience. We're looking here at what soil biological indicators you're already measuring in your work. So whether you're in Malaysia, whether you're in France, whether you're in Spain, which biological indicators are you measuring to show biological health? The poor arthropods, no one cares about them. They are on a big fat zero. Earthworms, everybody loves an earthworm. We do a lot on earthworms too. They are easy. You can find them and you can learn lots from the different types. Fungi, great to see that one rising up. Microbial biomass carbon, bacteria. Okay, we've got a few arthropod fans in the house. That's good news. Great to see that. Nematodes. I buy a lot of those on the internet to help with pest control in my back garden. Very, very useful too. Wonderful. And next question, what do you use those biological indicators for? Is it just for your own personal interest? Is it to help inform farm management plans? Is it for reporting up to your supply chain, to government? Why are you counting arthropods, earthworms, microbial activity? Soil carbon dynamics, guide to progress, soil indicators, field monitoring. Yeah, we see a lot of farmers really use this as a way to see that their soil health is returning, is restoring, and understand maybe what changes you might make to your rotation, any changes to tillage or amendments you might add to your farm. Testing regen practices. Yeah, again, really, really important. So many exciting new practices in the world of regenerative agriculture, but which ones are actually working? Nutrient release, yeah, pollution, erosion risk. I think so important. As the weather changes, having really healthy living soils helps us fight the kind of encroaching, changing climate. Wonderful. Thank you all very much. So next up, we've got another brilliant presentation, this time from Dr. Maeva Labourie. I think I got that right. She is a project officer for soil biodiversity at the European Commission Joint Research Centre. She graduated in France as an agronomist, but specialized in data science and holds a PhD in soil ecology from the University of Zurich. She really focuses on data analysis and modeling to address ecological questions and to contribute to the soil biodiversity component of the EU Soil Monitoring Directive. A really, really exciting new law that came in just, a month ago, that's going to make this stuff happen even more. So, Maeva, real pleasure to have you here. Over to you.
Many thanks for this introduction. So I'm here today to share with you the soil biodiversity assessment across Europe we've realized with the Lucas Soil Biodiversity dataset. And the first thing I would like to share about soil, but I think you're very well aware from this, it's that soils are beautifully diverse. They are full of life. This life can take all the shapes and forms you can think of, from the infinitely small with the archaea, bacteria, fungi, some protists that are example of microorganisms, up to tardigrades or nematodes for the mycofauna, columbolas for the mesofauna, and earthworms as our very well-known representative of microfauna in our backyard. But all this living diversity is involved in many soil processes, from carbon and nutrient cycling to organic matter decomposition to the structure of the soil formation. If you think about the earthworms and their burrows and how they transform the soil below our feet. And all this below ground life is strongly interacting with the above ground world as well. If you think about the plant-soil interactions through positive ones like symbiosis with arbuscular mycorrhizal fungi or a nitrogen-fixing bacteria that are helping the plants to uptake their nutrients. And on the opposite, we also have some parasitic behaviors with, for example, fungal plant pathogens. And all these processes, soil processes, are directly linked to many different ecosystem services with supporting, regulating, provisioning outcomes that are all at the basis of several food security pillars. And here, in the meantime, soil life has a central key role in all those ecosystem services, but is also subjective to environmental and anthropogenic changes that deeply shape it. And among them, we can find climate changes with extremes in temperature and precipitation that are even more expected in the future. You also have pollen management practices leading to deforestation, degradation, soil pollutions, and all of them are deeply changing the soil biodiversity status and are major threats. Especially land management practices have been recognized by experts as being among the top threats to soil biodiversity. And all these pressures in turn affect the ecosystem services that soil biodiversity supports, with direct and indirect consequences on soil health and also on our human health, as you've seen with the presentation, how everything is interconnected also with gut microbiome and so on. And here, soil biodiversity, as mentioned, is a key natural resource as itself, also at the central core of many ecosystem services on which we rely as a society. And yet, this resources faces multiple threats that we cannot fully grasp yet. And there's a clear urgent need to understand current patterns of soil biodiversity and the driver of their change so that we can preserve it by first establishing a reference point now of soil biodiversity status that we can compare in the future and understand how soil life responds to ongoing pressures. However, while we have large scale of soil physicochemical properties for decades now, the equivalent for soil biodiversity has not been matched. Soil biodiversity has not been included in international soil samplings for a long time, and this gap has somehow prevented us from assessing its current status, monitoring its evolution, and by extension prevented us from establishing and taking action about its preservation. And so while the importance of soil biodiversity for ecosystem functioning and soil health is now widely recognized, even more with the EU soil monitoring directive finally adopted. And this recognition, has not been matched by large-scale data collection efforts yet at the EU scale here. So to fill this gap, we need a large-scale understanding of soil community patterns across ecosystems type, soil types, climatic conditions. And here we can build on existing experiences from the soil physical-chemical properties assessment that have been existing for quite some time. And among these, we can find the Lucas Soil Survey, which is particularly useful in the sense that, so it is part of Lucas, that is the Land Use Cover Area Frame Statistical Survey. And Lucas Soil itself includes 20,000 sampling points, open access protocols and data, sampling across semi-natural to intensively managed areas, so from broad-leaved forests, coniferous forest up to annual rotation crops. And the sampling is done with standardized soil collection. And then the soil physical-chemical properties there are assessed with ISO standards for reproducibility. So building on this, as of 2018, soil biodiversity component has been added to the Lucas soil. That is called the Lucas Soil Biodiversity. And to date, this module represents the largest European soil survey that we have with harmonized open access data. Because all samples are processed by the same laboratory for soil biodiversity measurements here, and they are also taken according to standard operating procedure for the soil sampling. For Lucas Soil Biodiversity, the protocols and DNA sequences are available now in open access, and dataset covers 881 sampling locations across EU plus UK, UK being a member state back then in 2018. It spans 7 different land cover types, and in each sampling location, soil samples are collected for DNA metabarcoding of archaea, bacteria, fungi, and other eukaryotes, up to macrofauna. In this presentation, I'll focus on 2 major microbial groups that are bacteria and fungi. But there are more works that have been done here. So here, before going too much into the detail of the result, I would like to present some key notions so we can all match what I'm about to explain. The first thing is that DNA metabarcoding can spot genetic individuals that are called OTUs. And for each sample, we count the number of times a certain OTU is detected. And by doing this for all samples in all OTU, you can have an OTU table on the top left. That's what I'm working with most of the time, where the cells represent how many times that OTU has been seen in that sample. And for each sampling location, we also have some information about that location, like the land cover type it belongs, cropland, grassland, woodlands, and so on. Measurement of some soil properties like pH, climate, carbon, nitrogen, information about climate as well. And OTUs as well have their identity card, let's say, because they can be identified taxonomically by being compared to taxonomic reference databases that list several species, phyla, and so on. And for each OTU, they can be identified taxonomically, so down to a certain level of precision. Some of them will only be flagged as bacteria, some of them just flagged as fungi. And further, we managed to go down to the species level, or at least right before the genus. And this is stored in a taxonomy table. And finally, there are functional databases that are able to link an OTU to a function, like nitrogen fixation, based on its identity taxonomically, such that in the end, what I'm working with is we can tell for each sample who is in there, who they are to some extent, and what they potentially do. In the incoming slide, I will talk about richness, Shannon diversity. And those 2 notions are quite important as well to present here quickly. The richness is the number of OTUs that you detect. For example, in the sample 1 on the top left, you have OTU 2 and 3 that are detected, so the richness is equal to 2. And the Shannon diversity, while for the richness, we do not care exactly how many time an OTU is seen, so the proportion of one OTU compared to the other. For the Shannon diversity, it's really important to see if the OTU is represented in a quite balanced way compared to other OTU within the same sample. And with this in mind, all this data, we've published two different outcomes. The one that I will present is the first one you can see, the pattern in soil microbial diversity across Europe, focusing on bacteria and fungi. But there's also a very good work from my colleague that is focused on the 18S part, so all the fungal communities and other micro, meso, and macrofauna. So here in our paper, the main objective was to infer continental scale effects of vegetation cover, climate, and soil properties on soil communities to understand the environmental drivers shaping them and their patterns. And on the more anthropogenic side, we also explored how the richness and the diversity would evolve along a gradient of land use intensification. And for this, we analyzed bacterial and fungal DNA sequences from 715 soil samples collected across EU plus UK in 2018. And this sampling location also included both semi-natural and highly managed environment, meaning that we ranged from conifers and broadleaf forest up to intensive and extensive grasslands and permanent crop and non-permanent crops. And we considered that woodlands would be less disturbed environment compared to grasslands, themselves less disturbed than croplands, with the most disturbed environment being the permanent crop with these annual rotations. For each sampling location, we also had information about physicochemical properties and the climatic contexts. And one first thing here that we discovered through this Lucas Soil Biodiversity module is that we first considered the OTU richness, so the number of different individuals detected per sample. And we look at their distribution along this gradient of land use perturbation from woodlands to grasslands to croplands. We did the same thing with the Shannon diversity that is not shown here. But what we found is that both bacterial and fungal communities were richer and more diverse in grasslands and croplands compared to less disturbed environments like croplands. And so here you can see for the fungal example, where you have richer communities in grasslands and croplands. What we did here as well is to look at the functional profile, meaning that we also related the OTUs to potential functions based on their taxonomy. And what we found is that the communities that are richer, so on figure A on the left, are also the communities with relatively more potential fungal plant pathogens. And it's pretty important because it means that by just considering community metrics like richness and diversity, without further insights into the functional profile of your communities, we would miss the fact that, yes, we may have richer and more diverse communities in the disturbed area, but it doesn't necessarily translate into improved ecosystem functioning because we also have potentially more pathogenicity into agricultural areas, which is not really the purpose. So the first finding here is thus that both taxonomic and functional diversity need to be considered together to get a fuller picture of the communities that we have. Then behind those land cover types that are associated with soil properties, climatic conditions, vegetation, we wanted to see those three main environmental drivers, the impact they had next to each other on communities. So not interacting, but just additively considered. And what we found is that in our models, bacterial richness and diversity were both better explained in terms of patterns by soil properties and how they change between location, while for fungi it was more by vegetation. And we also found that the same soil and climatic conditions could show opposite relationship between functional groups, even if those groups are from the same organism types. For example, looking at AMF relative abundance, so avuscular mycorrhizal fungi, we found that they showed, for example, a negative relationship with the temperature seasonality, so how diverse are the temperature across the year, how changing they are. While for the fungal pathogens, these very unstable temperature conditions were positively related to them, so they like this type of chaos. And while both of these functional groups are part of the fungal communities, they showed completely distinct relationships to the same stimuli in terms of environmental condition. We also wanted to see as is happening in nature, how the interactions between major environmental drivers would permit to better explain, potentially, the patterns that we see in bacterial and fungal community. So we looked at two-way interactions between soil properties, climate, and vegetation again. What we found is that these models were better explaining our patterns, but also more informative. Because, for example, when looking at the most influencing interactions driving the presence of fungal plant pathogens, we were able to differentiate the effect of pH under different climatic conditions. And what we see is that in any cases, the proportion of fungal plant pathogens, the potential proportion, always increased with an increasing pH. More pH equal potentially more fungal plant pathogens. But this increase is even more important in contrasted temperature conditions. So they are worsened. And this type of analysis could permit to break down large areas like EU plus UK into smaller patches of influential soil type, climatic context, and vegetation. So we can better understand what's happening at a smaller scale based on this large scale analysis. I'll wrap it up here with some take-home messages about the fact that I think now it's more obvious that we need to take both taxonomic and functional diversity into account when approaching communities, even if it's based on potential function. It's already a fast first screening of the type of population we have. The fact that both bacterial and fungal communities respond differently to the same stimuli in terms of environmental conditions, and even within the same organism type, we could have functional groups with varied responses. And finally, the fact that considering drivers' interaction between soil properties, climate, and vegetation could permit to also take action in combinatorial patches of those drivers and scale down also the approach that we propose. And I will end here this presentation with a beautiful snapshot of how diverse the soils are. Here you can see. And I love this picture. You have all the references here on the top right. A part of them are part of the Global Soil Biodiversity Atlas. There's also the great site Chaos of Delight. If you ever want to see more about soil organisms, highly recommend it here. Thank you very
Amazing.
much.
Thank you so much, Maeva. An amazing presentation. And just in case you thought this was going to get easier, this topic, Maeva, I think, has shown us that there's so much complexity. Even when we start to measure these things, it's hard to know exactly why we're seeing the patterns that we're seeing. And there's often, things pulling them in opposite directions. pH, temperature, land cover, vegetation type. Yeah, Mother Nature is a wonderful but complicated thing to get our heads around. So over to you all. When you're thinking about using soil biological indicators, what barriers are there to you using them? Why don't you do more of it? Why is it something that you struggle maybe to convince your colleagues of? Why is it that soil biological indicators often slip down the pile? High variability. Yeah, we've heard about those from those first two presentations. The cost. Yeah, you need trained professionals often to get out there to see this. Not many labs do it. I think that's very true. It takes time, noisy data. I think, yeah, you only had to look at Maeva's graphs and there is, tens of thousands of data points. And how do you get the kind of the analysis through there? Brilliant. Yeah, complex. There's not a commercial solution to do it. We're teeing you up nicely here, Sonia, for your presentation.
I like what I
Perfect.
see.
And then another quiz question before we get to Sonia. But how long would it take to decompose the most abundant organic polymer on Earth, cellulose, in soil without enzymes at 25 degrees Celsius? Few days, few months, hundreds of years, millions of years. Hundreds of years seems to be taking the lead at the moment, but millions of years is making, oh no, it's coming up. Few on the months, and not very many people think it's just days. Got a few more votes coming in. Yeah, it looks like the hundreds of years is the clear winner here. So Sonia, handing over to you. You're a soil scientist, a biotechnologist driven by a passion for design and innovation. Some of those challenges I'm sure you just saw flash up are the things that get you out of bed in the morning. And for the last 4 years, you've been the Chief Technology Officer and a co-founder of a Swiss startup called Digit Soil, where you're all about developing practical tools to make soil biological assessments easier. So real pleasure to have you here. You're making this stuff easier and more possible. So looking forward to learning more.
Fantastic. Thanks a lot. I was really happy to see that the challenges that were mentioned, that I'm going to talk about them today. Do you see my screen well? Is it good for you?
Perfect. Yeah.
Fantastic. So, as you can imagine, it is always in science, it depends, but there is a reason why we keep our records on paper. Cellulose is extremely stable. It can be billions years to have a half-life of the cellulose before it decomposes. And that's, if you want to read a little bit more about it, I recommend the second publication on my list. They go in depth into the topic. But what I want to go into is what actually decomposes cellulose. What decomposes everything in our soil is enzymes. And it's such an important part of the biology of soil, and it's so often overlooked. But what I also want to do is to lead you a little bit from the soil biological indicator to wider interpretation, what we can do with these indicators and how we go around it. So how we think about soil biology at DigitSoil, since we are a company, we think of it that you need to make something that is practical, something that is usable. And how do you do that? You've seen on your pool already, Lure also mentioned it, that analysis of biological parameters is difficult. So what we started with, it was like, hey, so why don't we make it easy? So let's make measurement easy. So anybody can do it. So you don't need fancy lab, you don't need a lot of money, you don't need trained professionals or a little bit trained professionals to actually execute the measurement. You have to start with data, you need to have something to work with. And then, of course, then you come up with all this data, and then to make it usable for anybody who actually works with soil, you need to process it, you need to interpret it, you need to put it in a context. But still, for making it useful, you need to take one more step, which is taking it into actionable context. And this is what we've been working on to make the from sampling to action path easier. And I want to take you with me on that path today. And if I don't cover something, please reach out to me afterwards. I'm happy to have a further discussions. But like you probably guessed from the question I have put before my presentation, we are focusing on soil enzymatic activity. So what is soil enzymatic activity? Very briefly, you see here schematics. It's a food web. We know it's complicated. Yes, there is a lot of things living in the soil. But at the end of the day, all complex molecules, everything big has to get smaller for plants to take it up. For everything to cycle, you need to break big molecules into smaller molecules. And this is what enzymatic activity is. This is done by soil enzymes that originate from different sources. So they can originate from plant roots, they can originate from earthworm casts, so poop of earthworms or anything else that lives in the soil, but they're also produced by fungi and bacteria in order to do exactly that, to mine nutrients, to break big things into small things. So it's driven by enzymes, and that's a huge topic, but I'm going to focus on specifically hydrolysis, extracellular ones. So the enzymes that reside in the soil, that's been released or they sit on the surfaces of living things, they are in the soil. But so I'm not sure if you ever think about it that way. I want to put a picture in your mind if you think enzymes are proteins. So this is how we imagine proteins now. So there's a model. It's strings that are twisted and they move. So that's why enzymes are a bit should be considered something active. It's not a parameter that you just say, oh, that's it. They are moving elements of the soil. And what they do, if you can see here, there are little Pac-Mans. They would go and have very specific parts in this complex molecule that would attach to big molecules in the soil, and they will make them smaller. And these smaller molecules, then they will move on. That's why we think of soil enzymatic activity as a dynamic parameter. It's not how many enzymes there is, it's how fast they are releasing things into the soil. And this is crucial for us to understand how fast nutrients are being released in the soil. Therefore, it's always thermodynamic parameters. So we always, when we think of enzymes, we look at the time and we look how fast, how much of things is being released over time. Let's say glucose molecules from the cellulosidase is released over time. There will be slope of that curve. And if you can imagine, it's already very difficult to do. So every enzyme in soil will target different molecule and it will release something else. And to measure it, you would have to measure every single molecule that is being released, which is a lot of work. It's difficult. So there is a trick to do it. You can take chemicals that pretend they look very similar to the molecules that are present in the soil that enzymes normally target, and you attach a little additional chemical to them. So when enzyme in the soil meets that molecule, a fluorescent product is released. And by fluorescent products, I mean basically molecules that emit light if they are shined on, meaning that we can actually see them. And it's a fantastic method that allows us to be very sensitive to see which enzymes are acting in the soil, but it makes it also a potential. So we give artificial molecules and we feed the soil enzymes with these molecules and we measure how active they are. But here once more, we come to the problem that measuring this, it's a very annoying process. I've done it many times. You need to, with classical procedures, you need to take your soil sample, you make a soil slurry, so basically a soup. So you mix your soil in a big bucket, it's very low, and then you have to put it into the plates like this, many replications, many times, and add chemicals on top. It's manually complex. It's easy to make errors. And then you need a big machine to read your samples, and you need to wait. Since I said this is a rate, so you have to measure every certain interval of time to get that curve, to understand, okay, after 1 minute, I got 3 molecules, after 2 minutes, I got 6 molecules, and so to collect enough of the signal, you need to wait. Therefore, to make it easy, we thought, okay, no, we don't want to do that. We have thought, okay, let's make it simple. How do you make it simple? I have here the reader. I don't know if you see because I don't see myself. So I have a photo, but I also have it in my hands. We came up with this soil enzymatic activity reader. So a device, it's now being published. So if you are into very much details on how this has been validated within AI for Soil Health project has been validated, the results from the devices, you can go directly there. I will just briefly explain how it works. So it's the device that is independent of laboratory. You don't need scale, you don't need anything specific, you just need this, and you need a spoon. But the true heart of the device is, you see it on the slide, is the little, on my hand, we call it reaction plate. And I have one also in my hand. I don't know if you will see it. It is really that small. And what we've done, it is a standardized ready thing to use. So we have packed all these chemicals I mentioned before that would react with soil enzymes and emit light into that system. So it's ready to go. You can use it right away. And we have already all these 5 enzymes that are the most commonly measured ones, we put it in there. So if somebody is interested in their carbon or nitrogen or phosphorus cycle, how fast the enzymes in their soil are acting, they are covered. It's like a bit of a fingerprint. And And I have a little video that my colleague made to illustrate how the procedure is to make you understand how easy it is. So that's the device that of course you need to plug it. Then you need to put your soil into a little box that we used to call cuvette, but I think we have to change the name. And then that's the reaction plate that I'm holding in my hand that is sealed, so it can be stored, can be shipped. Then you just have to put it on your soil, press it, and put it into the device. So there, the device addresses the two problems. The one problem is the easy of use and processing of the sample, the time needed to process the samples, and all the errors that can be done in the pre-preparation, but it also analyzes the data. So what you can get at the end is the final result. So enzymatic activity for all these 5 enzymes that are already included in there. So that was our idea to go forward to bring the soil activity indicators to people, practitioners, but also laboratories. Obviously, the traditional method can also be replaced by this one. But this is only the first step. The second step is what to do with it. Like, how can you actually turn it actionable? And very often in the literature, in science, you would see graphs like that. And please don't go to sleep when you see them. For example, here we have the beta-glucosidase enzyme, and then it's being averaged over different ecosystems. So it can be very interesting insight into how different enzymatic activity group in different ecosystems, but this is not very actionable. So since enzymatic activity is driving processes, why not think about it like it's a process driver? So here is another publication that I wanted to show you is enzymatic activity against how much nitrogen has been released over time. So I encourage you to start thinking about soil enzymatic activity not as a status of the soil, but as something that can drive the soil. And then if we can think about enzymatic activity as a process that a number that helps us understand how fast things are being released, then we can start to put it into the actual agronomical context. So we, like Lure was showing, biological indicators as enzymatic activity is one of them as well. They are slaves of thermodynamics. So they will be more active when it's warmer, but then maybe drought will limit them. So you need to have a context for interpretation of this result. So you need to have a weather data, you need to understand if there are nutrient sources in the soil, and you need to have a farming context. And one application that we specifically are working on is to understand if and how can we use soil enzymatic activity to predict the amount of nitrogen, soil nitrogen, released by the enzymes to the soil throughout the season. Because we know there is a certain needs for plants to grow over the season, and then there is a question of how much fertilizer needs to be added to reach the certain yield. But the big unknown of that equation is how much your soil is providing. Therefore, we are working on building that application specifically. We already have, well, I'm originally from Poland, so I have a My Polish Garden here, where we have an example of how we could display this, where we can really directly say, hey, your crop needs this much, and then your release potential of the soil is this much. So you need to add the missing part only. This could be very powerful to limit losses of fertilizer or adapt the timing if we know that due to weather there would be more or less soil mineralization. So I think I went through the whole path, I hope, at least. So we have been developing the simplified system to be able to analyze soil, to provide biological activity data, and then have the data processing engine that digests the raw data into the soil enzymatic activity data, but also puts it into the context And then we also work with the extension, so farmers and advisors, to actually how to plug it into practice. So how to help to manage that resource, which is soil and the nutrients that are already in there. And with that, I have this couple of take-home messages that please don't forget soil enzymatic activity is important, it's there, and that's with it we can get potential rates of how fast things are being decomposed in soil. We are here also to show the easy-to-use standardized method that you can use anywhere you are, and propose that soil enzymatic activity should be utilized now once we can collect the data very easy to model soil nutrient release rates in the context of farming. And with that, I want to thank you for listening to me. And here is my team from Digital. I also want to thank them for all the work and developing the products together. Thank you.
Wonderful. Thank you so much, Sonia. Amazing to see how you're making all of this science of the soil biology easier and really useful for farmers. So thank you for that. And you can see we've taken you on a bit of a journey today. We first had a presentation from Lure that was about the deep science. How do you measure this stuff? What underpins which soil biology and biological indicators are doing what? Then Maeva showed us a kind of implementation at a kind of continental level. What patterns do we see if we start to do these tests across Europe? And Sonia's gone down another level and shown us what happens if we do this at the farm level. How can this be useful for farmers in helping them understand how much nitrogen to apply, how to make their Polish gardens more productive or their farms? And it's at this end, the kind of farmler implementation, that we at Soil Association Exchange work. So Soil Association Exchange is a company that I set up about 4 years ago. And what it does is it helps farmers to really understand how their practices, the things they do on their farm, are impacting the environment. So we look at biological indicators of soil health, but also chemical and physical ones. We look at biodiversity indicators, birds, plants, biodiversity connectivity. We look at impacts on water, and we also look at impacts on emissions and carbon sequestration too. And with all of that information, exactly like Sonia said, we help turn what is effectively lots and lots of data points into insight for a farmer. They can understand which bits of their farm are at risk of climate change, or which bits of their farm could be more productive if they could get Mother Nature working harder for them. And so we help them make an action plan of things that they can do to improve. And we link all of those actions through to money for the farmers. We help them unlock financial incentives. And I think what's amazing, maybe the biggest change that we'll start to see in this industry is, as Maeva said, the EU soil health monitoring law. That's going to come down the line. It's going to put a requirement on farmers to start thinking about these things. Companies that buy from farmers are increasingly going to have to think about whether their supply chains are resilient if they're working with farmers that are thinking about this stuff. And what all of that means is there's opportunity for farmers, opportunity to get price premiums from the companies they sell their produce to, opportunities to build a more resilient farm. We've found it to be a really attractive offer in the UK. We're working with about 2,000 farmers across the length and breadth of the country. And yeah, through the service, 90% of them, they do change how they farm, and 80% think they are more profitable, that we've helped them find a way to make money from farming in a way that's better for the planet. Super. So at this point, we're going to hand over to you in the audience for your questions. I can see lots have been coming in to the Q&A box, so keep those coming in. We might not get through all of them, but we'll send around some bits afterwards as well if we don't manage to answer all. But do scribble your questions into the Q&A box, and we'll see how many we can get through in the next 20 minutes. So first question, and maybe I'll come to you, Lure, for this one. Do we know which farming practices improve key soil biology factors the most? And how might we create an iterative agricultural policy that supports these?
Okay, so I would say there is no magical solution, and it depends a lot. In my experience, I have recently participated in a project where we were analyzing the impact of different sustainable practices in several countries, and it depends a lot on the context. So there is no magical practice, and my advice would be to test it in your soil to see if it works or not. But apart from that, for example, organic amendments are one of my favorites because they are like gold for soils. They provide nutrients, they provide structure, they provide biodiversity, or they support biodiversity. And I think at the policy level, more and more initiatives are supporting. For example, the last CAP policy or aids are testing their and measuring the effects of the practices that they support, if they really have an effect on soil health. So I think more and more policy is making use of the evidence and making tests to really evaluate if the practices they support are being effective or not. So I think we are moving towards there.
Super. So no magic bullets, see what works in your context, but sometimes there are some organic amendments that can really help boost some of that activity. Nice. Maeva, a question for you. If a member state of the EU or a country was looking to integrate soil biology indicators into their national soil monitoring program, what indicators would you suggest are the most resource effective while useful for understanding soil health?
Well, thanks for this question. That's a very good one. For now, I would answer based on the EU Soil Monitoring Directive that is using DNA metabarcoding for bacterial and fungi. So why not first going into this direction of bacterial and fungal communities? But actually, the bioindicators is a very interesting question because that's something which is very much under discussion at the moment. For bioindicators, you need some standards to assess them, some harmonization in the way we can have something coherent between member states at different scale, national, EU scale. So it's very much under progress at the moment. I cannot give you again a magic list, magic answer, as said by Laure. It's something that needs further discussion on that. We're actually slowly organizing a symposium in order to discuss such methodological standards to maybe hopefully reach a consensus on which bioindicators would make sense in this idea to have something comparable between different scale, infranational, national, EU, and so on. So stay tuned for the
Stay
rest.
tuned.
But...
Nice. So it sounds like maybe emulate some of the stuff that's been happening already. There are some practices that people are already doing, but what we really need to see is some standardization, and that work's kind of ongoing. So stay tuned.
Yes.
And maybe a linked question for you, Sonia. It sounds like a lot of the things that we are discussing today could potentially be really expensive for farmers. I'm always amazed, in more of a kind of physical property of soil or chemical, but, soil organic matter measurement can cost farmers tens of thousands of euros. And though it's important for them to understand it, maybe they don't see that value back, at least in the short term. And so how do you think we balance the kind of cost of doing this stuff well to inform practice at a farm level, but also not getting almost too obsessed with the science and spending so much money on measuring it that there's not enough left to support farmers actually on the transition?
Well, I can share my opinion on this. Maybe unpopular one. I think there is a little bit of a conflict of interest between the policymakers or high-level monitoring versus the farmer's interest. Sometimes it can be. Therefore, does the farmer really need every parameter of their soil and pay for it? That's probably the answer would be not. I would say probably if we want to really focus on the practitioners and people that work with soil, we have to really focus on what they need and find actionable. And for research and soil protection, we need to have another funds that would fund that part that we need to make policy. We cannot just shovel it to people that already spend a lot of money and effort on their food production. That's their livelihood. And we cannot just say, hey, now for our soil monitoring law, you need to do this, and pay all of that from your own pocket. So I think I would say we have to be quite aware and reasonable to bring people what they need and also collect the data that we need for making policy decisions, but to not just put the costs on them.
Yeah. I think that's a really good, I like this kind of two-tiered approach almost that, it's clear that there's maybe been a lack of focus on doing the deep science around soil biology indicators, and we need dedicated money to do that well. But that should come from governments and research councils and things like this. And then there should be a kind of parallel track, which is much more affordable, pragmatic for farmers, and maybe the two interplay a bit, but we can't push the cost of more expensive soil monitoring onto farmers without, helping them, see some of the financial upside. Maeva, we've got some questions for you, a bit more specific on your study. How have you defined a fungal plant pathogen? How did you, I guess, decide which were the good ones and the bad ones for the purposes of your study?
Okay, so and I think I also saw a question on how they were implemented, right? So it was all using DNA metabarcoding techniques. So based in the case of fungi on the fungal, the full length ITS region. So it's a target region that you go for identification in the metabarcoding. And in that case, it's the full length one. And based on the taxonomy, so this identity of the fungal OTUs, you can relate it with a functional database, in our case, fungal traits. So for each OTU, based on its taxonomy to a certain level of specificity, this functional database has potential function associated to that taxonomy and then hence to the OTU. And that's how it has been implemented in our case. It was not a choice based on our good or bad option, which is very subjective and anthropocentered then. So we went for this functional database. There's fungal traits. There are also other ones. Fungal traits is the one that we went for. Based on this, in that functional database, it can be also opportunistic pathogens that are mostly saprotrophs and that in the right or worst condition can also become fungal plant pathogens fully. Also saprotroph, but yes.
Super.
So in this case, it's purely on DNA metabarcoding, and that's why I was mentioning that it's potential functions and the first fast screening of the community functional profile.
Wonderful. Thank you very much.
Thank you.
And then maybe a question for you, Lure or Sonia. We've got a few questions that have come up in the chat and in the Q&A, I guess, about some of the kind of the limitations of some of these biological methods. So, someone said, Sonia, about enzymatic activity is, best observed almost straight away when you take it out the soil. And Lure, you touched on, how temperature and rainfall and pH can impact these things. And so what kind of practical advice would you have for someone that wants to kind of get involved with these measures to deal with, I guess, all the contextual stuff that makes this really complicated? So how can you have rigor in a scientific method, but deal with the, I guess, the reality that Mother Nature is complex and all this context stuff is there?
Do you want to go, Sonia, or? I would say that basically you have to take that into account. So if you are measuring these properties at the local scale, try to do them all under the same conditions, and that would, yeah, you save this situation like this. And maybe in the larger scale, if your monitoring will take a longer time periods, maybe this can be a disadvantage. And as I said before, maybe in that case, some of the properties would not be ideal for this kind of purpose. But yeah, I think it's when we do this kind of analysis, we always annotate which were the conditions when we did the measurements, and that's the most important thing to help you interpret the results afterwards, basically.
Nice. So almost collecting all of that stuff with it at least helps you, whether the number is good or bad, you've got the context to help you explain why that might be. Really good advice. Sonia, anything to add?
Yeah, well, for us, when we work on a very local scale, so not the European scale, but for a local scale, the options are also that we support, so geotagging your samples, so supporting the very precise collection of the samples, and also providing of the context, like Laura said. But also, if we speak of a multiple years of monitoring of a smaller unit, give it a field, a farm, we also obtain then historical data that can help us to interpret or spot the errors or see discrepancies. That even if the execution is not perfect each time, we can see patterns and try to like improve the process, because we cannot expect everybody to be a scientist. So if we want to go practical, we also have to have a bit of a different approach on how we look at the data.
Super. Nice. And maybe a final question for you, maybe before we open back up to the Mentimeter. But this whole series, this project from Horizon is about AI for soil health. And a lot of actually what we've heard from you today is much more around kind of physical sampling. It's the need to, get your hands dirty and count worms and fill up enzyme discs and things like this. And if that's the way that you do soil health biology well, what's the role of AI in extrapolating this stuff out? And do you think it could ever have a kind of role in being able to do this more affordably at scale?
So I'm thinking about the usefulness of mapping soil biodiversity at large scale. This can be done using modeling. This can be done using specific ensemble models and more like refined models where you do not only look at linear relationship, but more complex one. And using this type of modeling to map soil biodiversity could help also having a broader overview of their patterns in areas that are not sampled yet. I still think that the sampling on site is crucial in the sense that you can verify what is modeled. It's also on the soil properties, for example, that you can have a real action in terms of land management practices and so on. But I still see both approaches as complementary. One to take action, the other one to be able to predict based on what we have thanks to the soil sampling in other areas.
Yeah.
Especially in that case. So to me, it's purely complementary, and mapping can be one outcome of that.
Super. Yeah, it's often something we think about, that going back to the question we had about cost earlier, the physical stuff is expensive, but it does give you the gold standard in terms of what's happening right there.
Absolutely.
But maybe the gold standard isn't necessary if you've got really good models that give you the silver standard in other places.
Yes.
And, it's about having the right data for the right job. I think that's a really nice way to look at it. So we're going to throw back to the Mentimeter for one final question for you all in the audience. We'll just load that up. But if you find that screen again on your laptop. So the closing question is, after today, all the amazing things you've heard, what biological indicators would you like to try using when it comes to soil health? Are there stuff you've heard that you'd like to give a go. Some things you've heard, oh man, I'm going to do that on my farm this afternoon because you've really piqued my interest. What kind of things are you going to give a go? It's going to be a busy sales week for you, Sonia. This is good news. Soil enzymes everywhere. Some words, arthropods. They've been way too neglected today. It's great to see them finally getting their place. Metabarcoding. Yeah, there's some great companies out there doing eDNA. We've done a bits on farms in the UK, and you can learn so much from those tests. So really exciting to give it a go. Great. Well, I'm going to come to each of our panelists one final time, just to give us, if you've got one key takeaway for the audience here, if they only remember one thing from today's webinar, what should it be? Le, we'll start with you. What's your one takeaway for the audience?
As a scientist, I shouldn't be so biased, but I would like to finish saying that yes, soil biology is important. That is very relevant and sensitive. So we should try to implement them at the policy level, but also working with farmers. We should try to use soil biological indicators more and more.
Perfect. Yeah, I think that's so key. If you just take one thing away, it's soil biology is really important. Do not neglect it. Yeah, thank you. Maeva.
Well, I can just agree with Lo on that for sure. The fact that it has made it through the first soil monitoring directive is already a huge achievement that I see as a scientist here, outside of my link to the GRC. I'm very happy to see this because there's a great work from one of my colleagues, Julia Koeninger, on in defense of soil biodiversity. I think that's the paper, how it's called. And you could tell from many decades that we've recognized the importance of soil life and soil biodiversity. And still it wasn't matched yet. And I'm very happy today to see how many progress we are going for. So I can only agree with Lo that biological indicators are needed, and we're actively all working on it here at our different scales. So
Super. Great. So it's really important. It's coming, right? It's already here. You're going to see more of this. So you need to get on the bus. This is coming to your country, your business, your farm. So now's a great time to get involved. And Sonia, your takeaway.
Well, I would say I of course agree. Laura and Maeve already said the key points, but what I would add on top is that do not be afraid to measure soil biology. I think, yes, we are at the beginning, but I think it's worth doing and trying and collecting more data, and to really link it to interpretation and to make use of it. I mean, please come up with new methods, please measure more, and always talk to people that want to need it for their livelihoods. Then they have a lot of insights already and they can tell you what exactly they need so we can actually work for them.
Yeah.
That
I
would
think
be.
that's such a great closing message. I think one thing's for certain, having listened to all of you speak so brilliantly today, it is not simple, this topic, but that we make sense of this complicated thing by giving it a go. And it's brilliant scientists like the three of you that have come up with processes and tests and studies that have helped us understand. And what we need is more people trying more stuff at the farm level, at the government level, at the organization level, because it's then that we'll really start to understand how this complicated thing really works. So that's a wrap. Thank you all so much for coming. It's been a pleasure to have such a engaged audience from all over the world. We will be uploading a recording of this webinar so you can watch it back. If you've got any questions, feel free to send them through to us, to our panelists. All of our contact details are in the chat. And we'd love to get some feedback, what you thought of the event, what you thought went well, what you'd like to see more of in the future. As I said at the beginning, today was the last webinar in the Envisioning the Future of Soil Health Monitoring. But AI for Soil Health will be back with more information, more events, and all of that in the new year. So please do connect with us on Twitter, on LinkedIn, so that we can keep you up to date as those things come out. Thank you again for joining us. Enjoy the rest of your day.